can start introducing me.
Okay, so are you ready for our last keynote? Okay, guys? Okay, so Professor Neil Lawrence is Professor of Computational Biology and Bioinformatics at the University of Sheffield. He was in Manchester before moving to Sheffield, and he's going to talk about systems biology, gene networks. And again, this is one of these titles, like Magnus' one, where it's a free title. He will free to say.
He'll elaborate within the
exactly.
Yeah, I guess I couldn't remember what title I'd actually given Marta, so I made one up. Okay, so I guess I have the job of dealing with you guys when your brains are completely full, and you're very bored of R. So you'll be pleased to hear that there is no R in this talk at all. So I want to really sort of give you some motivation for, I guess we take a particular view of systems biology, and what I want to do is try and give you some motivation for that point of view, that perspective in this talk. And I want to use an example which is target identification, but I think it's a wider, it's an example that has wider implications, and I really want to focus on the relationship between computational biology and systems biology, and how these two things can perhaps come together. So, start with a bit of motivation, and then this is a little bit technical, so I'll go through this fast, not just because I don't think it's really important to get the principles of what's going on. So I guess the paper that maybe lays out the case for systems biology is this Can a Biologist Fix a Radio by Leibniz. And it makes a case for systems biology. And this is my favorite quote in it, which is this one, it's difficult to find a black cat in a dark room, especially if there is no cat. Now, I have no idea what that phrase means, but in some sense that also reflects my understanding of systems biology at some points. So, but I think the message in the paper is that biological systems are immensely complicated, and what Leibniz is arguing for is the need for models that are quantitative. So by quantitative models, we want sort of models that should be predictive of biological behavior, but then crucially, I think we want models that can be combined with biological data. Now these three things are sort of not quite in the fact that the system's complicated, but we need predictive models of behavior. And when I say behavior, I mean within the cell. And we also want models that can be combined with biological data. Unfortunately, these things somehow work against each other, because the more complex the system, the more complex the model has to be, the mathematical model. And when I say model, I will always mean mathematical model. I think sometimes people use the term model just to mean a representation that they feel there's some interaction. So I'm talking about a model where you've specified what the interaction is in a mathematical way. And then this sort of third point, that these models need to be combined with biological data. So this is problematic because, okay, if the systems were simple, then we could make predictive models that could be combined with biological data. With complicated systems, if we didn't have to combine them with data, we could still make models that were predictive. It would just be difficult to see how we could integrate the data with these models. So systems biology is really the way I see it, or what I think when I hear systems biology, I think it's associated with sort of systems engineering. So what I would claim it is building mechanistic models based on the biochemical knowledge of the system. So trying to write down a mathematical representation of what's going on. At its most basic level, people are using chemical equations to represent what's going on in a cell, trying to represent each possible interaction between elements that are interacting in the cell, like a transcription factor and the DNA, etc. And then the idea is to sort of identify modules and submodules and parameterize these models. And the reason for that is this idea of identifying modules and submodules is that Leibniz argues that it's the approach that people have to understanding how a radio works. It's not really fixing a radio, it's understanding a radio. If you look at what it is in biology, it's actually to go in and sort of destroy a section of the radio and then see what effect there is on the radio. Actually, the radio will just break. So reverse engineering, I mean, that's not quite, that's a sort of simplification, because actually what really goes on is, what you're really trying to do is perturb the system in some way. So you go into the radio and you sort of put a signal in at some point in what you've identified as a module. But you can see inside the radio that there are these different components of the radio, and you can sort of see, oh, well, if I perturb this, I expect it will help me understand this part of the system. And it's important to do that in biology, and indeed people do. They're trying to describe what the modules and submodules are. But there's a big difference between a radio and a biological system. A massive difference, because the radio was designed by a human and was actually designed to be quite simple. There's very little robustness in a radio. If you knock out a component in a radio, it's likely to not work after you've done it. Whereas it's amazing in biology, the number of things you can sort of play with and knock out and destroy, and actually the system still works. And that's because the system's evolved. And it's robust to these sort of perturbations. So if the system wasn't robust to these perturbations, it would have died out. So biological systems have lots of redundancy in them, which means that the process of reverse engineering them, even if it was easy to actually go into the system and start perturbing things, which is much easier in a radio to start putting signals inside the radio. It's difficult in biology. There's a lot of experimentation. Even if that stage was easy, the fact that there's multiple systems trying to perform the same role actually complicates matters a great deal. So I think there's a question, which is has systems biology been successful? I can't get to these things now. There we go. Has systems biology been successful? And I would say so far it's not yet delivered on the promises that it's made. But that's perhaps always going to be the way when something has become, there's a lot of money pushed into something and a lot of people promise things to get hold of the money, and then it doesn't necessarily deliver. But I think it's still important and it's still the right thing to do. And the way I'd like to argue that is actually going to computational biology, which is really what you guys have been looking at today. And I'd say that computational biology for the biologist is currently a much more useful technology. And when I say computational biology, what I mean is the sort of things you've just been trying in R, which is doing things like using gene expression data to speculate on co-regulated genes. So you've got some gene expression data, you load it into R, you process it using Puma or whatever, and then you do some sort of clustering. So there's a very traditional approach. And when I first sort of saw, the Michael Eisen's early work on this, I think hierarchical clustering is also a really nice thing to do. I mean, from a methodological perspective, which is where I come from, it's not perhaps the most interesting, exciting method one could use. But from the biologist's perspective, it's an approach to trying to get genes that are doing similar things together and visualize an entire structure. So you can look at it and say, oh, I think that these things, I know these things from the literature are co-regulated by the same transcription factor. So what about something else? What about the things that clustered with those guys? Are they also co-regulated? And this is sort of computational biology. There's very little modeling of the system going on. I'll explain what the modeling is, what the assumption, why this works. I mean, in some sense, when you consider the complexity of the underlying system of the cell underneath this, it is amazing that this works, because the number of things that cell could possibly be doing in terms of the complexity of the interactions between transcription factors and DNA and RNA and translation and everything is tremendous. But it does work, and I think one reason it does work is it's reflective of the wealth of new data that's coming in and our baseline ignorance about these systems that we're looking at. So what you're pulling out with these clustering is very obvious things that all these genes are moving together. They're all co-regulated. The subtler things, which we'll talk about in a moment, that aren't going to emerge from doing these types of clustering. So the sort of process I'm arguing for is one whereby it's an iterative process, where at the beginning, you're trying to do, well, you do a sort of high throughput experiment such as gene expression. So you perturb your system in some intelligent way as a biologist. You then measure perhaps gene expression over a time course to see what the response to that perturbation is. And then you do some sort of clustering, some analysis in R like you've just been looking at. Now this then suggests something. This suggests a bunch of targets, co-regulated targets. Now the next stage is actually to build a slightly more complex model, one that, as I said, I'll explain what the model behind clustering is, to add extra components to that model which are slightly more mechanistic, and then go in with a more refined experiment to perhaps look at one module that you've identified as being activated during the first experiment. So the sort of summary of what the way I view the world is that there's these two approaches to handling the data, what I think of as modeling. And what you've been looking at as computational biology, I think of as data modeling. And what a lot of what's going on in systems biology, where people are writing down very complex interactions between the different components in a network, I'd say is mechanistic modeling. And my argument, the point in these two humps here, so this bump here, is supposed to represent how much activity there is in each of these areas. So the argument is that there's a lot of activity going on in data modeling. The computational biology side, people are very active in that, coming up with new algorithms for analysis. And there's a lot of activity going on in mechanistic modeling, which isn't necessarily always tied in with the data because the data is difficult to get hold of. Some of it is. But actually what I'm sort of want to argue is that there's a big gap between these two approaches, and that gap is the thing that we need to, as not you guys, but as a computational biologist, that I'm interested in filling. So in data modeling, the idea is you introduce very few assumptions and you sort of let the data speak. So you just have your gene expression experiment, and then you do some sort of simple analysis, well, simple from a modeling perspective, principal component analysis, cluster analysis, hierarchical clustering. Standard sort of thing that you guys should be doing. In mechanistic modeling, though, you do much more. You sort of impose physical laws. You say, well, I know that the way you get mRNA is that you get RNA polymerase binding with DNA, and that leads to an mRNA of that gene, and then that leads to, and then you model very detailed in what is going on, sort of down at the molecular level, saying 1 TF plus pol2 plus DNA leads to 1 RNA plus pol2 plus TF, because things debind. Very explicit. And there's a whole language called systems biology markup language for being very specific about what you're saying in these systems that people use. So in data modeling, you tend to think of computational models like also support vector machines as well as PCA. And here you're trying to model in terms of systems and be more explicit about what those systems are saying. So here, I think that these, well, the way I term these things technically is adaptive models. So ones that can respond to data. So they're flexible, they see data, and they adapt to the data. Differential equation models is actually the sort of standard, or difference equations in some cases, standard approach to modeling in this side. So it's interesting, if you start showing a differential equation, people immediately assume that you're in this area here. So if you say, I'm going to model this system with differential equations, people will assume you're in that area here. If you start saying, I'm going to model a large amount of data, so I've got 30,000 genes across 200 experiments, people will assume that you're going to be in this area here. And again, the argument is that you want to, well, I start, and Magnus as well, from this direction and want to move there, and I'd argue that the people who are in this place want to be moving in that way as well, so that we can actually get what people need, which is a smooth move from high throughput computational biology to systems modeling, and that idea of actually being able to specify, ideally mathematically, how the cell's working. I mean, that's a sort of pie in the sky goal perhaps, but it's still there. Okay, so I term this weakly mechanistic because actually there are assumptions underlying these models. So it's not so explicit, and I want to sort of highlight that in a moment. So there are assumptions, and so I think of these models as weakly mechanistic, whereas these other models, they're much more strongly mechanistic. They're very explicit about what the assumptions underlying the system are. Okay. So we're advocating this hybrid approach between systems and computational biology, and I'd probably be repeating some of the stuff Magnus has talked about, or maybe some of the stuff Nicolo's talked about. I think that's all good. But it's part of, so Magnus and I both are sort of in this area of being interested in trying to bridge this sort of gap. So I want to use I hate actually quoting biological examples to biologists, but here you go. This is the example we'll use, and for me it was an inspiring example. It's work by Martino Barenko and Mike Hubank and collaborators at the UCL Institute of Child Health. It's a really nice data set. It's gene expression in P53. So just the background to it is that I'm assuming you probably know a lot more about P53 than me. But you've got radiation damage in the cell, and most of that's quickly repaired. But double-strand breaks are more serious. So the response of the cell is to, well, P53 responds to double-strand breaks, and it's responsible for pairing DNA damage and activates DNA repair proteins, also pauses the cell cycle to prevent the replication of damaged DNA, and it initiates apoptosis. So it's got these sort of, I guess, well, I'm sure experts in the area would say something much more than this, but from my computationist perspective, it's got these sort of three roles that I know about, and it also works in a sort of large-scale feedback loop with NF-kappa B in order to try and kill the cell if it basically has been damaged too many times. So I think the classic example I always think of is in your skin. As you get older, if you're exposed too much to the sun, you have a lot of dead cells, and the reason is because P53 has been activated too many times in those cells, and apoptosis has occurred to prevent them becoming cancerous. So yeah, I like these pictures of it because I just think that this one here looks like the flying spaghetti monster. I have no idea what all this stuff means, but it definitely looks like the flying spaghetti monster. And then that's it bound to the cell on the right-hand side. And then I even like this one even more, if it comes up. Because I mean, as far as I know, we don't know anything about this happens. It's amazing me how, I mean, so this is like an idea of the P53 binding the polymerase, and I like that stuff. I just like looking at those pictures, even though I'm sure that's nothing to do with what really happens in the cell, but it's a fun drawing that someone's done. Okay, so this is a very typical situation that the sort of experiments that they did was to perturb a system, a cell line system with some radiation, and they have some knowledge. So they know that these five things are sort of well-known targets of P53. So there's DDB2, which is DNA damage-specific binding protein. So that's, I guess, to do with repair. There's the cycling-dependent kinase inhibitor. So that's to do with, I think, pausing the cell cycle. Also, this cestrin-1 is cell cycle arrest. And then there's a couple of things that are involved in apoptosis or necrosis, these two down here. So you've got two involved in the cell cycle, two involved in cell cycle pause, two involved in inducing cell death, and one involved in perhaps DNA repair. So here's a simple module network, and this is what Martino and his colleagues suggested could be going on. So the idea is you've got the transcription factor P53, and he's governing these 5 known targets. So we've got some knowledge about the system. And what we're interested in is if we have this knowledge, what can we say about other potential targets of P53? Now, this is an experiment you could all do. You just simply go in, you perturb the system, in this case by radiation. You take a time course to see how the system is responding, using some knowledge about the length of time at which these processes occur. And then you do clustering on the resulting time course. You then use your knowledge of these guys being known targets, and you look for clusters where they appear. And you say, well, things that are in those clusters could potentially be other targets. So it's a nice sort of setup. One can do that experiment. And so what I want to sort of motivate is what the next stage would be, which is making that experiment a little bit more complicated. So the standard approach is to assume that co-regulated genes will cluster in the same groups, then perform clustering on the data set, and look for those clusters that contain the target genes, and then speculate that those are potential other targets, then confirm by other means any ones that you might find interesting by looking in the literature or further experiments, and then publish. So it's a nice sort of setup. It's what I think of this as a spiral of research, like a corkscrew going in, driving in to find the knowledge. So there was one stage which was there's the system, bottom one looks at a system, then there's the experiment, and then you move around to a little bit of modeling, very simple clustering in this case, which suggests another experiment. Now, what I'd like to argue is that actually that should keep going around and round and around. At some stages along that spiral, you publish, but you're sort of screwing this corkscrew in, driving forward, learning more and more about the system you're interested in. I'm not saying this is what always happens. I'm just saying that this is what I think an ideal model of would be. So, as I said, Martina Barenko and Mike Eubank, they actually, the work I'm talking about is really their work. It's very much in line with what I think. But they did this experiment using this, what they call the hidden variable dynamic modeling. But the key point about the experiment is that they don't just do clustering. So here's the main maths I want you to sort of look at and have a think about, which is this very simple model. And what I liked about this work is I'd wanted to work with models like this, but I didn't. My worry was, as soon as I said differential equation, people were going to think, oh, he's doing mechanistic modeling, but that's not the right mechanistic model. You know, he's not doing the modeling correctly, so he shouldn't be allowed to model. So this is a very simple differential equation, and what it says is that you've got this rate of transcription. So mj of t is the mRNA concentration over time. And so this term here, actually I'm going to end up having another slide, and this term here is the rate of transcription of mRNA-J. So that's one gene, yeah? And the rate of transcription is dependent on three things. Well, so or the rate of production in some sense, because we also have this decay term here. So this says that the mRNA is decaying. So over time, we expect that if there's no production of mRNA, it will just decay and disappear. And this is a simple so-called linear decay term. It says that the more mRNA there is, the more mRNA will disappear, basically, which sort of somehow makes sense, because if there's more mRNA, there's more of it decaying, and that's all that term says. But then this term, unfortunately, the green is maybe harder to see here, is the term that's dependent on the transcription factor. So you've got, well, in this one equation, you've got one potential target mRNA, mj over time. You've got one transcription factor of interest, in this case p53, p of t. So both of these things are functions. That's why we're writing them in this form. So you're going to maybe have measurements, but over time, these things will be doing something like this. This may be the p of t, and this may be the mRNA, mj of t. But both of them are continuous functions. Now, what you get, I mean, because actually what's going on underlying the system is a continuous amount. What you actually manage to observe is you might take gene expression measurements of just the mRNA. So we're assuming in this case that the transcription factor is difficult to measure because it's a protein. So it's actually quite easy, and you know, the standard approach to measure large numbers of gene expression, it's difficult to measure large numbers of protein expression, although you can, of course, using Western blot or whatever. But we're assuming that this curve here is somehow unobserved. What was the driving transcription factor? I shouldn't say concentration, I should say activity, because of course it's activated p53. So in this system, there's some p53 sitting in the cell that becomes phosphorylated due to DNA damage, and then it becomes active. So this is a representation, this curve represents how much active p53 there is in the cell. And that's, you know, not necessarily easy to observe. So, and then there's a baseline. So what the baseline says is even in the absence of p53, there may be some production of the mRNA. So this could be, what was that, BIC? I can't remember gene names. This could be any of those genes we talked about. This could be. So this is our model, that there's this rate of production of mRNA, which is given by some constant rate at all times, some sensitivity to the transcription factor activity, and some decay. Now, there are three parameters in this model. There's this basal transcription rate, which has to be positive, anything from 0 going all the way up. This sensitivity to p53, so a non-target, if it was 0, this would be saying, you could learn that this was 0, and it'd be saying, well, I'm not sensitive to p53. And this decay rate for the mRNA. So these are three. The point about these parameters, though, is whereas in clustering, and principal component analysis, there are, well, less so in clustering, but there are parameters underlying the data, they're not necessarily connected with any physical reality. Here, these parameters may not be connected with physical reality. For example, if the cell expands, then the concentration of the mRNA decreases. So it's not the actual mRNA has decayed, but that has to be dealt with by this parameter here. So these parameters are trying to deal with these phenomenological ideas, but the model isn't complicated enough to cover everything that could happen in the cell. It's a simplification. Is it a useful simplification? Well, what I want to argue is yes, it's a very useful simplification, and I want to argue that by first clearing that, and then rearranging this equation in this way here. So why do I rearrange it in this way here? That's because on this side we've got, these are the knowns. I mean, they're the knowns. They're not as well known as we'd like. And this is the unknown. So up to the parameters as well. But in terms of the functions inside the cell, we can make observations of the mRNA through gene expression. But we can't make, there are approaches, but it's more difficult to make observations of P of t. So somehow this P of t is unknown, but it can be expressed in this equation. What we've said is on this side, this stuff is known. Okay, if we knew the basal rates, the sensitivities, and the decays, then we could also reconstruct what this P of t was. Now, the system's a little bit more complicated than this because there are many mRNA. And in fact, in this case, we've got 5 mRNA that we're interested in. Not just one. So there's several equations like this, but in each equation, P of t is shared, and in the other equations, these guys will be different. Also, the sensitivity and the basal transcription rates will be different. So hopefully the idea is you can see somehow you could reconstruct what this transcription factor activity is. So what Martino and collaborators did is they estimated this production rate by fitting polynomials to the data. So what do I mean by that? Well, they had this sort of some gene expression measurements, and they fitted polynomial, which is a type of curve, to the data like this. And once you've got this polynomial, you can look at any of these points and you can say, what's the gradient? So you can say, this is mj over time. That didn't come out very well, sorry. This is mj over time. And then since you've got this curve, which is your estimate of mj over time, the idea was to fit that gradient. You compute the gradient of a polynomial quite simply, and that was their plan. The nice thing about that approach is it turns out if you fit that with a polynomial, that's also a polynomial. This is a mathematical thing. You don't have to worry too much about it, but I just feel like mentioning it. So the sum of two polynomials is a polynomial, and then you also, that means you represent P of t as a polynomial as well. And this was their trick. It's not the trick we'll use, but it's a nice trick that they used. Okay, so once they'd done that, they could jointly estimate P of t at the observations of the specific time points along with these guys. So they didn't estimate the entire function. They estimated P of t at the time points of interest, and they also estimated these parameters. I can't remember the number of time points, but I think it'll be clear in the following plot. But let's say it's 12. So if it's 12, then you have to estimate 12 activity times, and then plus each of these parameters for each gene. So there's 3 parameters per gene, 5 genes, 15 parameters. Oh, okay. So what's going on with clustering? Well, you can see with this equation now. So clustering is equivalent to assuming the decay, the basal rate, and the sensitivity are very, very large. What happens if you make that assumption? If these guys go very large, so they're going up, decay, sensitivity, and basal rate, they're going very high, this one has nothing in front of it. So as you make them large towards infinity, if you bring them up together, basically that term doesn't matter anymore. So if that's the case, you can then say, you can reorder this equation and say, we lose that gradient term, that one we had to estimate by fitting polynomials, or Martino and collaborators did. And you just have this linear equation. So all this says is that the mRNA we see, and if we bring this, you can bring this decay on the other side, so I cross it off there. The mRNAs we see is just going to be a constant plus a scaled version of the transcription factor. Now, if that's the case, that the mRNA, the transcription factor activity, if there are a bunch of mRNAs that you were looking at, and they're all just scaled versions of a transcription factor, that means they'll cluster. Because by scaled, what we mean is, so you've got one like that, they'll all look like this. So just different scales with maybe different offsets. And in a clustering algorithm, hopefully get rid of that scaling, and they'll all cluster together. And they're then obvious known targets. So in this situation, this is why looking at the model is useful, because it says what you're saying. It says that if targets are high sensitivity, so they've got a significant response to the transcription factor that's coming in, and they're high decay. So basically, they appear in response to the transcription factor, but they decay very quickly. So when the transcription factor goes, they disappear as well. Then clustering will work very well. And I think it just happens to be the case that that is very often true. There may be good, I don't know, one can speculate about why there are sensible biological reasons for that, but somehow that does seem to be often the case, that targets are high decay, high sensitivity, as we'll see for p53. Okay, so I'm having trouble with my keyboard. There we go. Right. So their results look like this. So they estimated for each of these 5 different genes, these known targets, what the basal transcription rates should be, given their gene expression, a bunch of gene expression data which I'm not showing, the sensitivity to the p53 activity, and the assumption here is that there's one governing transcription factor, and the degradation rates. And they did it across 3 replicates, so 3 replicates here, and then this is their estimate of the function. As I told you, they estimated only at the time points where they made gene expression observations. So this is their estimate of the function for the p53 transcription factor activity over time. And they've placed error bars here because they did a little bit of sensitivity analysis known as a Bayesian analysis. They also have error bars on these degradation rates, sensitivities, and the basal transcription rates. You will see that there's no error bars on the p21 ones. That's because there's what's called a scaling ambiguity here. So they fixed one of them to specific values to improve the identifiability of the model. So in some sense, you can see all these other parameters as relative to the p21 parameter. So you could see it as DDB2 is more sensitive to p53 than p21, and DDB2 has a lower decay versus p21. So these parameters are estimated as part of the algorithmic processes of estimating the model. And then they did Western blot to try and see, Western blot on what they feel is the active form of p53 in this case. And the profile's similar. It's not quite, I think those profiles dropped earlier. And they have some argument about why that should be the case, which is sort of biologically beyond me. So I won't repeat it. OK. So that's mostly about Martino's work. So I'd like to talk a little bit about what our work is in this area, which is actually to have what we call a probabilistic model for this transcription factor activity. So this is the missing function. And in my lab, we tend to, if something's missing, we use something called Bayesian Inference. So we place a probability distribution over the missing thing. So we don't know it, but the probability distribution could be a very powerful thing that considers all sorts of possibilities. It might include, and in this case it does, the probability distribution. It's over a function, which is quite a complicated thing to do. So the maths behind this, we don't teach it to any of our undergraduates or our MSc students. So don't worry about that. But the idea is that you're trying to incorporate assumptions about what you think that transcription factor activity should be before you've seen it. And so those sort of assumptions might be smoothness. So you might expect that it doesn't do that. So you can incorporate that assumption. I mean, someone can argue with you, say, no, it could do that. OK, but this is the assumption. And you're just explicit about what sort of assumptions you're going to use. So we do that through probabilistic modeling. Okay, so what I don't want to show you the sort of technical side of this. Come on. What I want you to sort of look at is these pretty pictures, which allow us to specify this probabilistic model. So you guys probably know about correlation, yeah, because you have to do lots of correlation tests. What we do to create these functions is we specify a matrix of correlations. And what this matrix of correlations says, so here's a function that's sampled from our probabilistic model. And it's only sampled, we can only show it at certain specific points. And what the matrix of correlation, this is the matrix of correlations between these points. So this is what we define in the model. So we put this into the model. That's from us. That's our decision. And what it says is that any 2 points in this plot, which we sample, are strongly correlated if they're closely related to each other in time. So when we say strongly correlated, that means that we expect, if we've observed one, if a point's strongly correlated with it, we expect it to be close by to it. And that's represented by these numbers which represent correlation in this map. So these are the time index. We've labeled the n, but you can think of it as a time index. So this is time point 1, time point 2. And this map here says time point 1 and time point 2 are strongly correlated. Yeah? So the correlation coefficient's almost 1. But time point 1 and time point 25, not correlated at all. So there's no correlation under our model between 1 and 25. There happens to be in this plot, but that's just coincidence. That's a statistical thing. There's no, if we sample it many times, we'll see that there's no correlation. This is just one instance. But what we know, if we sample many times, these 2 points will always be close together. Now that's quite a cool thing, because this model, we're just specifying the correlation between neighboring points. And then we sample from the model, so it's a probability distribution, we can sample from it. The way we specified the correlation leads to these functions, nonlinear functions, which we hope might look like transcription factor activities. And so this is what we specify. It's called a Gaussian process, and it's what a lot of my own sort of mathematical research is about. But it's a really nice object, because whereas here we're specifying the correlations discretely, and you can do that, that's one thing you can do. What we do in general is we specify these correlations continuously. So that's just reiterating what I said there. And this has got a nasty equation in it, but don't worry, you don't have to understand the equation. I mean, just to say that the equation is what's specifying this matrix. So we said discretely, but here we've just put time inside this. And the way we compute all these elements, this is just, you put 2 times in, and it gives you a number out. And the way it's developed is that if we ignore that alpha, this number can always be interpreted as a correlation. The alpha actually just gives the overall scale. So don't worry about the actual formula, apart from the fact that it's operating over continuous time. So what's really good about these models is we don't have to care if people have done discrete time steps, or people have said, oh, we did 3 time points close together, and then we did 5 time points much slower. A lot of methodologies developed in engineering for systems identification believe that, or rely on a fixed time interval between experiments, and can't handle it if it's not there. That's because if you're analyzing a radio, you can sample what's in the radio at a fixed time interval very easily. It's not a problem. It may be much more problematic. It's because there's no cost associated with making those samples, whereas in biology there's a lot of cost associated with each experiment. So people might want a more intelligent design where there's more samples taken around the period of most activity and fewer samples taken when the system's less active. So this system allows that. And what you can do is you can sample from this model of what these transcription factors might look like. So there's a few samples here. Okay. What's rather fun, and again you can ignore that equation. Just look at the pictures. What's rather nice is that differential equation we showed earlier, that simple linear differential equation, can be combined with this model. So this is what we're looking at as our belief in correlations between 2 points for that transcription factor activity, something we don't observe. That's the thing we specified in the previous plots. But the differential equation says to us, well, if that's true, And if we specify some sensitivities and decays, and here I've done 3, one's high decay, high sensitivity, one's medium decay, medium sensitivity, one's low decay, low sensitivity. And what the model says, if that's true, then you should see correlations of this type. So the high decay, high sensitivity will be highly correlated to P of t. That's the clustering example. So there's this strong correlation between these two, the high decay, high sensitivity, and the driving transcription factor analysis. That's what you're taking advantage of when you're doing clustering. But in the cases which are medium decay, medium sensitivity, low decay, low sensitivity, look at the correlations. They become much weaker. There's still structure there, but a lot of the structure is correlation through time. So if I try and draw a line down the diagonal, you'll see it's asymmetric. There's more correlation with older time points than there are with future time points. And that's a result of this differential equation model. What we'll do is we'll show you some samples from this differential equation model that try and highlight that. Okay. So this is sampling from that joint model. So this is our model of the system. This isn't real data, but what we can do is we can say, give us functions from that model. And what we get here is in black is a sample of what it thought might be an interesting transcription factor activity under our model. In red is the high decay, high sensitivity. So that's tracking it almost exactly. Let me just wipe that off. Come on. So the high decay, high sensitivity is almost following the black line almost exactly. Very strongly correlated. But look at the green line. This peak here, this top peak here, is sometime after this peak here. This is the medium decay, medium sensitivity. And the low decay, low sensitivity is even more different. The point about a plot like this is if that's the situation, it's a low sensitivity, low decay target, you won't pick it up in a cluster. So you won't identify, you will identify red as a target. You won't necessarily identify green and blue. Now, green and blue may be rarer cases, so that's okay. But when you're starting to drive down a little bit into your model to sort of say, oh, what are the other targets? Are there any missing targets? Then you might want modeling like this. You might definitely want modeling like this if people have looked at the system before, because if you find some new targets that are in this regime, then that's likely to be a paper. Here's another example from the same model with the same parameters. In this case, we've got a more wavy transcription factor activity. Again, red follows, green and blue follow less well. I don't know why keys aren't working here. Okay, there's another example. Again, same pattern. Okay, and now what I want to show you is what this means in terms of experiment. So we can actually fit the parameters, these sensitivities, decays, and basal rates given an experiment. In this case, I've assumed they're given to us. Normally, we would fit them, but I want you to illustrate how this type of modeling works in practice. What we've got here is we've got 3 potential targets, genes, from this transcription factor activity here. So the black is it's a simulation experiment, it's not a real experiment. So the black is our simulated transcription factor activity. And our 3 targets, one is low decay, high sensitivity. So look what it does. This is an integral of that function. So it goes up and stays up. It's responding to the transcription factor activity, but it doesn't decay. So the mRNA just hangs around in the cell. Here's a medium decay, so slow goes down over time. And here's the high decay, high sensitivity target that we're more used to, the red. Actually, you'll see the data come up, and I've put noise in it and tried to make it look like sort of gene expression experiments, make it more realistic. The parameters we're using in the model to infer on this side the transcription factor activity were learned from this data. I didn't give the model the parameters, I told it to learn them. But in this example here, it knows the parameters already. It did that learning already. So what you're going to see is as genes come in, they're going to look like these little dots here. So gene expression measurements from different time points at different genes. What I want you to observe is this, which is the estimate of the transcription factor activity. So at the moment, it's got no knowledge apart from we told it there's no activity at time 0 because we hadn't perturbed the system. As we introduce genes, you'll see this curve will change. This blue curve is the mean expected transcription factor activity, and these gray bars are the error bars. So it's very uncertain because it's not seen anything. But as it gets data, these error bars will improve and give you an idea of how good the experiment is. And I'll introduce the genes one at a time. But now I really get problems with my keyboard. So I'm introducing this blue gene first, one experiment at a time. Yeah. And after introducing the blue gene, what we see is it has some uncertain estimate of what's going on in the transcription factor activity. There's not enough information here for it to know because of the noise in the genes. So look how much noise I put on here. The noise here is actually about the same size as that bump. So it's no surprise that it's got a similar size bump here responding to that noise there. It doesn't know. It doesn't know that the system's flat because of the noise on the gene expression. But the idea is you add more information, that if that's a random perturbation, you'll keep seeing this feature, but you won't keep seeing that feature. So hopefully this sort of thing will iron out. So we'll introduce another gene. So as I introduce another gene, again with noise, look how much that bump flattened out. Come on. Look how much the bump flattened out. It was up there, but there's no equivalent feature here, or actually it would have to be here maybe, to say, that bump's there. But there is still the equivalent feature here. You still get that curve going on. So it still thinks something's going on here. And indeed, we get, I think, most information from looking at the third gene, the high sensitivity, high decay. But it's the information for these three genes in combination that lets us tell what the estimate of the transcription factor activity is. So what we're trying to say here is, despite the lack of information, just three genes which are known targets. That's the information we're putting into this system. Three genes which are known targets. We can make this estimate, and look how confident it's become. I mean, the error bars, it's a little bit out there, but they basically cover this system here. The model will always sort of hallucinate. It doesn't know about flat things. When you saw the curves we generated before, it's called a stationary model. So the model will always hallucinate these sort of things because it believes that the activity should keep moving. It's only data that tells it doesn't. So that's actually a slight flaw in the probabilistic model we're using over the system. But still, it will, as it sees more data, understand that that's flat. OK. So we published on this model on the same data from Martino Borenko with my colleagues Pei Gao, who she's now in Cambridge somewhere, and Antti Honkalo is a visitor from Helsinki who's still working on these models with us, Magnus, who you've already seen, and myself, and just using that P53 data. And these are the sort of experimental results we get. So this is the inference of the P53 protein, which also you saw for them. But notice for us, it's in continuous time. We infer it across all time. So not just at specific time points. And we infer it with these error bars. So that's the P53 protein there. These are some of the target mRNAs. Now, we used Puma for processing. So for each target mRNA, we have an error bar. If it's too small, I'm just drawing it in there. Here's another one for this guy. So what the model does is it says it's trying to infer what the actual activity was without noise. And so it's got a smooth line that's got error bars much smaller than the error bars of the observations, because it says that these things must all be coming from the same base source. So you're combining information from across these genes, which means that actually the error bars for each target gene are pretty tight, smaller than the noise was on the original experiment. So that's the sort of result we get for the target genes. And the idea is that you can take that sort of data and then fit that model to proposed future candidate targets. Now, when we do that, what we find is we have done that for P53, but I don't have those results here. Okay, so this is another example we did with collaborators in Manchester. The PhD student was Jennifer Withers. So this is an L1 system. And here, same setup. So here's the training genes. So these are the observed training genes. But notice one of the known targets has this low decay profile. So these are all combined together to have an activity that is that one up there. And then what we get is a predicted target gene from other potential targets in ELK1. So this is another potential target. It happens to also follow very closely high sensitivity, high decay. But the question mark is this a target? So it could be a target actually when at first glance, because it goes up early and then sort of does that. But the model says no. Well, it goes up early and then, so this is where the gene expression profile was like that. But the model says no because it's got this curve in it here and then there's another jump up here. And the model can't see how this profile can have explained that type of double jump. It may be a target of p53, but it would have to involve something else on top of p53. It's not a target of p53 only, of ELK1, because we're dealing with ELK1. This is a different experiment, of ELK1 only. So that's the sort of results we can get out of those experiments. So just to mention a final thing, one thing we did, which is a PNAS paper. This is with, again, with Antti Honkola from Finland, and then these are collaborators from EMBL, Eileen Furlong's group, myself and Magnus, was to actually include a model of translation. And the idea in this was that, okay, so if we have, so this doesn't work for the p53 system because phosphorylation goes on, but the missing thing is in the middle. This is the transcription factor. But if we believe that translation is the main way in which the transcription factor is being produced, and we have the mRNA of the transcription factor and the mRNA of the target here, then we can go through scanning the system. We're still inferring what's going on here, which is the thing we can't observe, but we can go through scanning the system looking for potential targets of different transcription factors. And this is what we did in this paper. So it's a slightly more complex model, but it's the same basic idea to use some mechanistic ideas inside a computational model. Oh, I've gone past it. Ah, okay. Sorry about that. And I have to do. Okay. So here's some interesting results in this case. So you can really see, in this case, what's going on. We've got actually another simple differential equation model of this system. And what you see is that you've got a driving input mRNA, this guy here, who is the mRNA of the twist transcription factor. And the question is this response possible from that mRNA coming in? And what the model says is, yes, that is a possible response. And the way it happens is that the protein looks like it's doing that. The transcription factor activity looks like it's doing that. And the response is to go up while that stays up. So with a low decay protein, you can get that sort of response. Now, interestingly, in that, all this model is looking at is those two guys, the mRNA of this twist transcription factor and the mRNA of a potential target. And it says the model fit is great. So it really ranks this very highly as a potential target of a twist. But interestingly, there are other targets that look like this. This is back to that high decay, high sensitivity regime. So high sensitivity, high decay protein, high sensitivity, high decay target mRNA. Now, it's not possible for twist to be working in both these regimes because these transcription factor activities should be shared. So the only way this is possible is if there's some modification of the transcription factor activity that we're not modeling for. But it poses a research question. Is twist regulating these two mRNAs in a different manner? The evidence from chip-on-chip is that it does actually regulate both these guys. So it's possible that there's some modification to the transcription factor protein, phosphorylation or ubiquitination or whatever, that actually causes this. But from my perspective, that's just speculation. I don't even know how likely that is. So there's a bunch of other targets there. So the results were, for this example, that actually in some cases we even beat knockouts in terms of transcription factor target prediction. So these are predictions from this model I've just shown you, the black, which is consistently very high. And this is for twist, and this is for another transcription MEF2. In particularly for twist here, the knockouts don't perform at all well in predicting the transcription factor as validated by chip experiments. So what we're validating against here is large-scale chip. And the chip is, we're much more predictive of what the chip experiments say the knockouts are. Okay, so just to summarize, what I've been arguing for is moving a little bit away from those computational biology models, introducing a little bit of mechanistic understanding to introduce what's hopefully a lot more power, and the sort of system that could be used in the second sweep of that spiral, or even indeed that last thing is something that anyone can apply to a time series. If it's developmental, that would be a typical correct application. And in fact, we have this as an R package called Decay. So other sort of ongoing work is actually to move more in the direction of those mechanistic systems by having more complex models. So we've got work on nonlinear response and nonlinear differential equations, work on improving the computational complexity. So some of these models can be quite slow because of the extra algorithmic computations. Something we'd like to look at is stochastic differential equations, and Darren Wilkinson does a lot of great work on that. He's a statistician in Newcastle, and much more towards the mechanistic side that I mentioned at the beginning. And then we also saw this cascade model that was making a richer model, had this translation model, which I didn't show the details on, but it's very similar to the transcription model. So just to collaborators, so I think on most of these grants, well, I used to be the PI on the grant we had, but now Magnus is the PI on all the grants. So, we often have grants together, and the funding for this has come from BBSRC and EPSRC, and actually we have ongoing projects, which I forgot to add, which are BBSRC projects with biological collaborators applying this stuff. Guido Sanguinetti was a postdoc who did some early work with this, and Michalis Tsitsias just finished with us. He's been looking at nonlinear differential equations, and of course you can't do any of this work without good data from helpful biological collaborators. So many thanks to Martino and Mike from the Institute of Child Health, and Charles Giraudot and Eileen Furlong from EMBL. That I should have mentioned, the last example was Drosophila melanogaster. And there's some references at the end. That's it.
So, are there any questions? Pretty advanced stuff, I would say, as a simple biologist.
Yeah.
And I guess from a utilitarian point of view, you present us with an analysis of a particular situation where you're looking at a particular transcription factor affecting some potential or known target genes.
Yeah.
When this kind of is fully developed and you've got a competent person like yourself doing the modelling, are you able to go searching for transcription factors that fit the model? In other words, because co-expression is a fairly weak tool in many cases, because relationships aren't simply about co-expression at the transcriptional level. So are you proposing this as a tool for searching for relationships that might actually help the interpretation?
I think that's a very interesting question. We've actually tried it. So we've actually tried to go to take mRNAs we're interested in and go through the known transcription factors, asking what are potential regulators. This was work with Marta and Matthew Holly. It's more speculative work because it's harder to do because it's needle in a sort of haystack. When you're looking for targets of P53, if you just say everything's a target, you're maybe 10% right or something. But when you're looking for a transcription factor of a known target, then it's only going to be 1 out of 30,000. And we haven't done any confirmatory biological work. But yes, that's the sort of thing I think we'd like to be able to do. And I think really importantly, and what I didn't talk about today, is to be able to combine data from different sources, which might help you find this stuff. So at the moment, it's down to the human to do that, using his knowledge of the system. But some sort of automatic combination of other types of information to help that process is something we're interested in as a research challenge, and we have some models for doing that in some systems.
So what would that data be? That data would
people have
stands
used
for
that, yeah.
relationship.
So we kind of do, in effect, we do use that type of data at the moment, not so much in this model, but yes, exactly that sort of data. But there's a human probably between the yeast-2 hybrid data and the model. So the human knows about the yeast-2 hybrid, so he believes these relationships are there, so he constructs a model based on what the yeast-2 hybrid information is. The examples where we're more directly looking at data are less associated with this type of study, but one of things Niccolo is doing is trying to combine, say, genotype information with phenotype, but phenotype in terms of gene expression. So if you've got sort of genome-wide SNPs, and then you've got gene expression, and then you want to start understanding networks through trying to combine the two. If there's a mutation that's affecting a particular gene, that sort of modeling is an area we're going into. But yeah, yeast-2 hybrid and whatever's on the web. There's a lot of work of people trying to search the literature automatically. That's very hard.
Question on development of systems biology model. A lot of development. These models are dependent on existing data.
Yeah.
And there's a lot of ambiguity. Or sometimes one lab, one group or a group of labs shows some data and the other shows completely contrasting data. So we don't know, is there a tool which enables us to convert the available data into a format that could be
Yeah, it's interesting, isn't it? So if two data sets are in conflict about what they're saying, then I guess one has to analyze what that means. So that could mean, I think that could mean a few things. Either someone's got something wrong, okay, but let's ignore that possibility, because I think the other possibility is much more interesting, which is they're in conflict because our current model of the system's incorrect. So there's something going on in the two different systems that we don't fully understand, and both things might be correct. That typically, I mean, that's actually sort of was coming through in the last thing I briefly showed, that cascade system, where two different models of the same system were saying different things about the protein activity. Well, it means, no, in that case it means that the model's wrong. It's a consequence of the model, the mathematical model, being wrong. But it's still informative, because it's informative if your mathematical model's wrong. And I think that that's why it's important to be very quantitative, and certainly from my perspective, the more modeling you do, the more explicit you can be about what's going wrong, what's the conflict between these two data sets are. I mean, if you're just saying, oh, one's going up while the other one goes down, I don't understand. You're not really saying, it's quite fuzzy. So my sort of motivation is, well, let's get the maths in there and say what it means in terms of a mathematical model.
I understand the whole purpose of developing a systems biology model is to enable us to do non-lab based, or what we call dry experiments.
I think people have said that. I think it's very like computers and paper, right? Xerox used to have this research center that was very worried about computers, because they said, well, no one will actually need paper anymore, because they're just going to use, this is in the 70s, and they came up with a graphical user interface as a way that people were going to operate. What was the consequence of computers? We needed more paper than ever, because people were printing things out left, right, and center, and you needed more photocopiers than ever, and there's been a massive boom in that sort of thing. I don't believe at all the arguments that say that there will be less biology done as a consequence of this. I think, if anything, there's going to be more biology done, because you can only do those sort of dry experiments in isolation if you are 100% confident about your model of what's going on. Now, we can be confident about sending a rocket to the moon because we believe that Newtonian mechanics is adequate to describe that process. Newtonian mechanics was derived in the 17th century, and we're now confident enough to do that sort of thing. What you're talking about is the equivalent of sending the rocket to the moon in the cell, and my confidence in any of the modeling we're doing is not anywhere near. I think they're tools, they're investigative tools at the moment. I know people have promised that, and maybe that's the Holy Land, but it's not. I'm not working actively towards that. I think there have to be intermediate goals, which is using model of a tool to improve the biology, improve the efficiency in which experiments are done. And that's the key for me.
Any other questions? You've demonstrated this approach looking at relationships between gene expression. Do you think there's a role for this approach in linking genomic data with, say, metabolomic data, extending the systems biology approach?
Yeah, I mean, it's a good point. Yeah, those two things obviously interact. I don't know how much modeling is going on there. One slight problem is I guess the model just becomes bigger as you do that. And I guess our instinct is to try and isolate parts and try and understand them. But I think you're completely right. This is a problem with biological systems in general because of this redundancy, that actually trying to claim that there's a module that just does this in isolation is going to be less true in a biology system than a system that an engineer's designed. So yeah, I mean, we're not looking at that, but I think that's an important thing too. Yeah.
A lot of the kind of systems biology that I'm familiar with is really revolving around what I would call micro modules. You know, they're just very restricted sets of elements that are cooperating in some kind of functional role. And actually, as soon as you go outside the domain of that model for those specific entities, it loses any kind of predictive power. And the problem is translating that to the scale of a system.
Yeah.
It has biological relevance outside that simple thing.
Yeah.
And I just wonder whether these models are going to be, I suppose, it's scale of computational capability to actually run these things simultaneously, and how well you can achieve the computation.
And also, I think that the interesting challenge intellectually for us is how you deal with the missing measurements. That's what really this is about. That you've got, if you could measure everything everywhere at all times, then maybe you're in a position then to bring a very complex model to bear on the system. And it just becomes an issue of computational complexity. But in most systems, you don't get to measure all these things at all times. And so then you're left with a large amount of uncertainty that you need to deal with. For me, that's a massive, interesting intellectual challenge, and that's what I really like. But I think that's another major problem for systems biology in general. You've got these missing things that you didn't.
Come through engineering and physics and maths.
But
You have a mindset, whereas a biologist might actually be prepared to just kind of take the rough edges and not try to analyse it down to precision, to precise ways that engineers tend to do.
But I think coming back to that promise to let's do systems biology and let's just do dry experiments to predict what a drug's going to do, I mean, it's not acceptable to not understand the details of the system because potentially there's some, well, and every individual is different. I mean, it doesn't even get on the fact that actually the more interesting thing I think that's going on now is we're starting to understand what the effect of a drug on an individual's just by characterizing their genotype. But not necessarily understanding the mechanisms, but just seeing, there's a group of individuals with these mutations who don't respond well. And I think in cancer that's very important too. But that's much more computational biology. But it still brings back this need to bridge the gap, I think.
Any other questions about it? Yes?
I have a funny one, but if you've got an experiment and you I don't know, you have just two contrasts A and B, and you pick out some transcription factors and cluster genes, you go, oh, this transcription factor might be controlling it. Could you go to either in the same species or another species, time series data, and actually apply that model? I mean,
it's interesting. So we're trying to do that with some. It's a really nice data set. I can't remember his surname. Alex Pavelnik. It's a Nature data set on Drosophila that we're looking at, which is cross-species, cross-Drosophila, and multiple different flies. One dominant data set from Drosophila melanogaster, which is also the Berkeley Drosophila database, which is the data we looked at, but all these different species. And their interest is looking at how these transcription factor activities or gene profiles have changed across evolution in different times. So you get these, but the really cool thing that we think is coming out is we're learning even more about the Drosophila profile as we see these other species. So there are these features that are sort of exploded a little bit in other species because that development phase is longer. And as you look closely, if you squint at the Drosophila data, you think, oh yeah, that's in there too. And I think that's really, that's a fascinating challenge. Yeah, and I think it's going cross species is a really cool thing to do, yeah.
Okay, thanks ever so much. I'm just going to wrap up today's work for a little bit. Can I just kind of talk to you as a whole about what you've got out of today and extend setting the processors up in your laptops and progressing with it. It's been pretty long. So, you know, can you tell me what you've got out of it?